All right, today we're going to talk about chapter 3, which is all based on cells. So there's a lot of information in this chapter, a lot of factual information, but also a lot of conceptual information. So it is a pretty in-depth chapter. So you want to spend quite a bit of time, memorizing some things, but then also really trying to figure out if you understand some of the bigger concepts. So why are cells so important? Our human body contains about 100 trillion cells. About 3 billion die and are replaced every minute. So this is a huge system within our body. Cells are also the simplest unit of matter. So we have cells such as bacteria, that can work alone, but the cells in the human body only function usually when they're a part of a larger organism. So we kind of had talked about some of the organ systems, and we talked about the organization of our body, and cells was a part of that last week. And now we're going to talk about really what cells are made of. So there's lots of different cell variations. Really why cells are in a variety of shapes and sizes has a lot to do with their function. So nerve cells, they have to be these long kind of cells to be able to get their signals where they need to be. Blood cells are usually circular. We can talk about some of the sizes of these. Most range in size from 10 to 15 micrometers, and a micrometer is 1/1000th of a millimeter. So we're talking about really, 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 really tiny cells and things. We're not talking about something that's usually quite visible. A blood cell measures about 7.5 micrometers. A human ovum or egg is 100 micrometers. So you can see that, that's quite a bit of variation in size. The human ovum at 100 micrometers is about the size of a period at the end of a sentence, so those are a little bit more visible. Nerve cells may even have extensions up to a meter long, so the length of a nerve cell can be quite large. Usually it's the size, like I said, that functions or that directs the function of the cell. You can see there that we have this gland cell. Its job is to secrete things. Talk about hormones, mammary glands, those types of things. Muscle cells, on the other hand, they have to be able to kind of stretch and then contract. That's how we have movement. So see why some of these things are shaped the way that they are. Let's talk a little bit about cell structure. So this is some of the memorization I was talking about. You're going to just really have to commit to memory the different pieces of the cell structure. I'm going to talk about almost all of the things that are listed here. The cell's basic structure includes the following. So we have a plasma membrane. So that's here on the outside. It really creates the boundary of the cell. We have the nucleus then that's in the center. We have cytoplasm that kind of is here in between the nucleus and the plasma membrane. And it's this gel-like substance that has a whole bunch of organelles and molecules in it, each of which serve, a very specific function. So we're going to talk about all of these different things here, including the plasma membrane and the nucleus. But what I want you to remember is that no cell contains all of these structures. We're going to talk about all these structures today, but really most cells don't have all of them in there. Specialty cells have certain variations of these organelles or molecules in them. First, let's start off by talking about the plasma membrane. So again, this really creates the boundary for the cell. This is a semi-permeable membrane. So do we know what that means? Hopefully we know that that means that some things can permeate it. So some things can move through, but it's selective. So some things can, some things can't. And it doesn't just let everything move across the boundary. And we're going to talk about how some of that movement happens a little bit later. This plasma membrane consists of phospholipids, cholesterol, and protein. So this is what we're going to see here on the outside. These are the phospholipids here. We have some cholesterol here, and then we have these proteins here. So those are some of the bigger pieces. They also sometimes have these carbohydrate chains on them, and those actually help to act as markers so the body can distinguish its own cells from foreign invaders. So they're kind of like marker carbohydrate chains, so our body can say, oh yeah, that's our cell. So the phospholipids you can see are the bulk of the membrane here. They have head and tail twins. So this is the head, and then we have these tail twins here on the inner aspect. The heads are what we call water-loving or hydrophilic, and the tails are water-fearing or hydrophobic. Phobia is kind of a fear, so maybe that will help you to remember it. They're positioned in this double layer. Some heads point towards the fluid-filled cell interior, right? So the inside of the cell here, and some point to the exterior. So that's what really kind of creates that boundary. The tails then point towards each other, and they form this hydrophobic core. So that's what allows this membrane to be selectively permeable. I want you to also think about this membrane as it's not stationary. This membrane's constantly moving in kind of a wave-like fashion, so it's very easily movable. It doesn't move really fast, but it does move, and it keeps the membrane kind of fluid. The cholesterol molecules are in here, and they help to stiffen and strengthen the plasma membrane. The proteins are embedded in various spots in the membrane, and they also fulfill many functions. Some pass through the membrane and act as channels, so this one here is kind of dictating that. It's going to allow molecules to move through the protein, acting as a channel. Some attach to the surface and serve as receptors for specific chemicals or hormones, so that might be this one here that's got this marker on the outside. And then other carbohydrates are attached to the outer surface, which we just talked about. These ones over here are just kind of attached, and they act as markers to help the body distinguish itself. So lots of different parts to the plasma membrane. The nucleus really is the most important part of the cell. Lots of stuff happening inside the nucleus. We're going to talk about cell division coming up towards the end of this section, and the nucleus will really play a role there. This is really the cell's control center, so it's kind of like the cell's brain. So we have a nuclear envelope here. It's really a double layer membrane. You can kind of see that depicted here. And it regulates the passage of materials, mostly RNA and DNA, outside of the nucleus. So it's important that we have this double layer and we have these pores. Most cells only have really one nucleus, although we do have a few. Liver cells and skeletal muscle cells are an example that contain more than one nucleus. Then we also have red blood cells that have no nucleus. So for the most part, all cells have a nucleus, but there are some exceptions to that. Extending throughout the nucleoplasm are strands composed of DNA and protein called chromatin. So that's what this is pointing towards here. It's really kind of this middle layer here. The nucleolus is in the middle of the nucleus. So it's kind of like a smaller nucleus. It manufactures ribosomes, which are the cell's protein-producing structures, and we're going to talk about those coming up as well. You can see here it shows you, sorry about that, it shows you that the ribosomes are attached here to the endoplasmic reticulum, which is also attached to the nucleus. So the ribosomes are going to be produced there, and eventually they're going to come out here and be a part of the endoplasmic reticulum. Just a point to note, this chromatin here is eventually which is going to become chromosomes during cell division. So we're probably all familiar with that word chromosome. So we're going to talk about that a little bit later too. Inside each cell is over 7 feet of DNA. So tons and tons and tons of DNA in each one of these cells. Eventually they're divided into individual molecules. But just to give you a little perspective of how much stuff is really inside the nucleus. So let's talk about a few of these other organelles that are located in the cytoplasm outside of the nucleus. The first thing I want to kind of talk about is the endoplasmic reticulum. So you can see here on this diagram, we have smooth endoplasmic reticulum, we have rough endoplasmic reticulum. We also see that it's giving you some description that this is, remember, the nucleus here, so the nuclear envelope, and this rough endoplasmic reticulum comes off of there, and off of that is the smooth endoplasmic reticulum. On the outside is the ribosomes. That's why this part is called the rough endoplasmic reticulum versus the smooth endoplasmic reticulum really has no ribosomes on it. Really, this endoplasmic reticulum is just a network of membranous canals and curving sacs throughout the cytoplasm. Like I said, the ribosomes are on the outside of this surface, and these endoplasmic reticulum and the rough endoplasmic reticulum really play a role in synthesis of proteins, and it helps to move these proteins and things towards the Golgi apparatus, which we're going to talk about next. So here's the Golgi apparatus, and now you can see that we have the rough endoplasmic reticulum here, okay? And what's happening is we get these proteins coming off of it via a transport vesicle going to the Golgi apparatus. Okay, so the endoplasmic reticulum delivers a protein molecule to the Golgi apparatus. The protein passes through each of the sacs of the Golgi apparatus, undergoing modification in here. So we're changing them, making those proteins whatever we want them to be. At the end of the Golgi apparatus, it kind of envelopes or creates this little package called a vesicle that contains a whole bunch of protein. This vesicle then moves towards the surface of the cell and actually secretes or releases those proteins outside of the cell so that they can go wherever they're needed. This is kind of just one thing that happens within the cell, but you can see how multiple of these organelles are working together to create these proteins that are then released outside of the cell and utilized in different areas of the body. Lysosomes really are membranous vesicles that form from pinched off pieces of the Golgi apparatus. So we were just talking about them. They contain various enzymes, and they serve a lot of different functions. They're kind of known as the cellular garbage disposals because they have these enzymes that break down proteins if we need them to. They also allow us to reuse amino acids. Amino acids are the building blocks for proteins. So we can have so many different proteins because we have a whole bunch of amino acids. They come together in a very specific fashion to form one protein. But think about it. If you had 10 different items, how many combinations of those 10 items can you make? Same thing with amino acids. You have a whole bunch of amino acids. They can form all sorts of different combinations, which create proteins. Lysosomes also play a role in destroying bacteria. So you can see that these kind of pinched off packages really do multiple things within the cell and outside of the cell. Centrioles are located outside of the nucleus, and they really look like these two bundled rods. I put a nice picture here for you, so hopefully see that a little bit. We're going to talk about these when we talk about cell division. Their primary role is to function in cell division, and you'll see them coming up in a little bit. Mitochondria, that's a pretty, common term, I would say. Most people who've taken any sort of basic biology have heard of the term mitochondria. They're, again, located within the cell, in the cytoplasm. There are differentiating factors. They have an outer membrane and an inner membrane. We call mitochondria the cell's powerhouses. They help to create ATP, which all cells use for energy. So that's why we call them the powerhouses. Back to these membranes for a minute. We have this outer membrane and inner membrane. The inner membrane kind of folds back and forth across its interior, and these folds are what are called the cristae, and you can see that it's where this is pointing, these folds that it kind of makes. The spaces between the cristae contain enzymes that the organelles use to convert organic compounds into ATP. So that's really all that's happening in the mitochondria. Mitochondria are present in most cells because we want cells to be creating ATP. We need ATP to use for many cellular processes. Cells that do a lot of work, such as liver cells, contain more mitochondria than cells that do less work, such as sperm cells. So you can see how this varies via what type of cell it is, how many mitochondria are you going to have, depending on that cell's specific function. The cytoskeleton of the cell really is the framework of the cell. It determines the cell shape, it strengthens the cell, it allows the cell to move. So you can see here, it's kind of like these tubes here, these microtubules and these microfilaments. It's really what create this cytoskeleton, and it allows the body to have cells that have a little bit of support to them. I want to talk about these three things at the bottom because you will need to differentiate microvilli, cilia or flagella. So that first picture there is microvilli. These really, all it does is it's an extension of the cell membrane, and it creates all of these folds that increase the surface area of the cell. So why might we want to increase the surface area of the cell? To aid in, movement of particles across the cell membrane. These are especially important in cells that need to absorb nutrients, like cells in the intestines, right? We eat, we break down the food, we want the body to then absorb those nutrients. So the cells in our GI tract are going to have these extensions so they can be continually absorbing nutrients. The cilia, on the other hand, are this next picture, which looks very similar, but these little projections do not increase surface area of the cell. They're hair-like cells that actually move. They beat kind of in waves to propel a substance forward. There's a couple of different places that you'll find these cells. The respiratory tract is one, helps to sweep dirt and molecules out of the respiratory tract that we don't want there. And then also in the fallopian tubes to push the egg down the fallopian tubes into the uterus. The third picture here is the flagella or the flagellum. These are really only occur on sperm, and they're meant to help the cell move. Okay, so you'll see that sperm, of course, needs a way to move, right? The sperm has to get up through the uterus into the fallopian tubes to meet the egg. So I've talked about the different pieces of the cells, the different organelles in the cells. I want to talk about how things move through the cell membrane. We talked that cell membrane, that plasma membrane, was selectively permeable, so it's going to allow some things to move back and forth. And then we're also going to have to force some things back and forth. So just to clarify a couple things, there's two ways that we can move things, or two overwhelming concepts of ways we can move things. Passive transport, which requires no energy. It includes diffusion, osmosis, filtration, and facilitated diffusion, all of which we're going to talk about. And active transport requires energy. So this includes transport by pumps and vesicles. Okay, this is often going to be going against what our body kind of naturally tells us to do, which is why it's going to require energy. So the first process of passive transport is diffusion. And diffusion really is a pretty simple concept. It involves the movements of particles from an area of higher concentration to lower concentration. It can occur in air or water. So this shows you a picture of a dye tablet. You put the dye tablet in, and as the molecules disperse, they're going to move away from where that dye tablet is because there's a very high concentration of particles there. Okay? What I like to think about is if you're in an elevator and it is packed full and the doors open, are people naturally going to move out of those doors? Yes, because they want to move away from where all the people are. People don't tend to want to be, shoulder to shoulder unless they're in a specific situation. When particles move from an area of greater to lesser concentration, as in this example I just gave you, the particles are said to move down the concentration gradient. Okay, so it's important we understand that term, concentration gradient. That means when particles move from a concentration of higher, from an area of higher concentration to an area of lesser concentration, they're moving down the concentration gradient. And diffusion continues until the particles are evenly distributed. You can see that here in this picture. I'm going to show you the animation for this. Let's try that one more time.
Diffusion is a natural movement of particles from an area of higher concentration to an area of lower concentration. Diffusion is a form of passive transport. That is, it does not require energy to occur. Diffusion occurs in air or water. It can perhaps be best illustrated by placing a dye tablet in water. As the dye tablet dissolves, the particles move away from the tablet where concentration is high to the edges of the container where the concentration of particles is low. Diffusion continues until the particles are evenly distributed. The point at which no further diffusion occurs is called equilibrium. Even when a membrane stands in the way, the particles will still diffuse through it as long as the membrane is permeable to those particles. A difference in concentration of a substance from one point to another is called a concentration gradient. When the particles move from an area of greater to lesser concentration, as occurs in diffusion, the particles are said to move down the concentration gradient.
Okay, so hopefully that helps to explain it a bit better. We're going to have lots of different animations for this because there is a lot of different concepts that we are going to go through. So the next one is osmosis. So we talked about diffusion, it's passive transport. Now we're going to talk about osmosis, which is also passive transport. And really the difference here is we're going to now talk about the movement of water instead of the movement of solutes or particles. Okay, so osmosis involves the diffusion of water down the concentration gradient through a selectively permeable membrane. Okay, so the water is now going to move instead of the solutes. So there's different examples of when this would move. When this would happen. If the semi-permeable or selectively permeable membrane does not allow for the particles to move, but allows for the water to move, well, the water is still going to move. We're still talking about moving down the concentration gradient. So think about when we're talking about osmosis, you're only thinking about water. And again, the water wants to move from an area of lesser concentration. Let me say that again. The water wants to move from an area of greater concentration to an area of lesser concentration. Okay, so let's look at this example here. Side A and side B. Side A has 5% albumin, side B has 10% albumin. So if you look at this, you can see less albumin here, more albumin here. If there's more albumin on the B side, that means there's less water molecules because this is at an even level. So the water is going to want to move over to side B because there's less water molecules over there, okay? So instead of talking about solutes or particles, which happens in diffusion, now we're talking about the movement of water. So let's look at the animation for that.
Osmosis is a type of diffusion that often happens when a particular substance cannot cross a membrane. The membrane in the container shown is separating a 5% albumin solution from a 10% albumin solution. The membrane is permeable to water, but not to albumin. In this situation, the water, not the particles, moves in an effort to equalize the concentration. This is an example of osmosis. Diffusion of water down the concentration gradient through a selectively permeable membrane. The concentration of the two solutions eventually equalizes, but in the process, one side ends up with a greater volume of water.
So you can see there how now it says at the top here 7.5% albumin, 7.5% albumin. The concentration of albumin in these solutions is now the same. See? Still less particles of albumin over here, more particles of albumin over here, but we have much more water over here. So the concentration is now equal. And that's what the body's always kind of trying to do, is get to that equalness. Okay, so tonicity or however you're going to hear people kind of pronounce it in multiple different ways. This is quite a difficult concept to understand, but eventually we're going to be talking about different types of IV fluids. I had a slight discussion on this a little bit with one of the groups on Thursday, but it's something that we need to learn about. So this might be something that you'll have to spend some time on. So let's go through some definitions first. Isotonic is a solution with the same concentration of solutes as that which is inside the cell. So we're always comparing outside the cell to inside the cell. So isotonic, a solution outside the cell, same concentration of solutes that's inside the cell. Hypertonic, a solution with higher concentrations of solutes outside than what's inside. Hypotonic, a solution with lower concentration of solutes outside than inside. Okay? And why this is going to matter is that this is going to play a big role in how fluids move, right? We just talked about fluids moving from a concentration of a higher concentration to a lesser concentration. So if the concentrations are different on either side of the cellular membrane, we're going to get some movement, okay? And I'm going to show you the animation on this.
Cells are essentially closed containers. Besides containing fluid, they also contain a variety of solutes, such as salts, sugars, acids, and bases. It is the concentration of these solutes in the fluid that determines whether and how much fluid moves into or out of a cell. If a plasma membrane is impermeable to a particular solute, and if the solute is more concentrated on one side of the membrane than the other, osmosis will occur. The three terms used to describe these solutions are isotonic, hypotonic, and hypertonic. A hypertonic solution contains a higher concentration of solutes compared with the fluid within the cell. If a red blood cell is immersed in a hypertonic solution, such as a concentrated salt solution, water will diffuse out of the cell, causing it to shrivel and perhaps die. A hypotonic.
So why does the water move from inside the cell to outside the cell, right? The water is going to move from inside the cell to outside the cell because there is much less water outside the cell, right? If there's a salt solution outside the cell, there's not that many water molecules out there. More water molecules inside the cell. So it's going to move to outside the cell. And then it's going to cause the shell to kind of shrivel up and dehydrate.
Solution contains a lower concentration of solutes compared with the fluid within the cell. If a red blood cell is placed in a hypotonic solution, such as distilled water, water will move by osmosis into the cell. This influx of water will cause the cell to swell and, eventually, to burst. An isotonic solution is one in which the concentration of solutes is the same as it is in the cell. When a red blood cell is placed in an isotonic solution, water moves into and out of the cell at an equal rate. As a result, the cells remain normal in size and water content. Isotonic fluids hydrate the body without causing dramatic fluid shifts.
So if we don't want fluid shifts, we're going to give somebody an isotonic solution, something like 0.9% saline or normal saline. Is going to do this. If we want fluid to shift in and out of the cells, then we're going to give a hypertonic or a hypotonic solution. So that's one of the reasons why we talk about this, is so that eventually we can understand those concepts when we start talking about IV fluid administration. So the last concept that I want to talk about for passive transport is filtration. You can read your book and kind of see the difference between diffusion and facilitated diffusion. That's the only other concept in your book. I do want you to look into that. I'm not going to talk about it because I think it's pretty straightforward, but I want to talk about filtration. So filtration is a bit different because filtration, water and dissolved particles are forced across a membrane from an area of higher to lower pressure. So now we're not talking about concentration of solutes or concentration of water. We're talking about pressure, okay? So naturally, there's a lot of pressure somewhere that is forcing kind of molecules or particles outside. So in the body, this happens often in our capillaries. Our capillaries force water and dissolved materials into the surrounding tissue fluid. It does that because we need to get these water and solutes and nutrients out to the tissue fluids. But there's a lot of pressure in the capillaries, so it does that on purpose. Filtration also happens in the kidneys. The kidneys, there's a lot of pressure inside the kidneys, inside each little nephron in the kidneys, and it forces the waste products out of the blood so that they eventually can become urine. One analogy that's used and talked about in your book is a drip coffee pot. So as the water drips into that container above the pot, as more and more water gets in there, there's a lot of pressure. So there's a small hole through the coffee grounds, the water is going to eventually drip through there because there's a lot of pressure where all that water is. So those are the processes of passive transport. I want to talk briefly about the couple of processes for active transport. Remember, active transport is going to require energy. So this is going to be a little bit different. Here's the sodium-potassium pump that I want to show you. And this requires energy because we're actually going to go against the concentration gradient. So instead of like in diffusion, where solutes were going to move from an area of greater concentration to an area of lesser concentration, in the sodium-potassium pump or an active transport, we're actually going to force molecules from an area where there's a lesser concentration to a greater concentration. So hopefully kind of in your head you can conceptualize that a little bit and it makes sense to you that that would require energy, right? So let's go back to that elevator. There's a whole bunch of people standing in the elevator. The door's open and people want to get on. That's going to require some energy, some movement of people to allow for those people to get in there. People aren't probably going to be really happy about that. They don't want that elevator to be crammed right full. So that might require some discussion or even arguments to allow those people to get on the elevator. Same thing here with sodium-potassium pump. We actually have to force these solutes to the areas where there's already a great concentration of them. So they don't really want to go there, which is why we have to use ATP. So let's watch the animation on this and we can see how that's going to work.
In active transport, solutes move up the concentration gradient, from areas of lesser to greater concentration. One of the most important examples of active transport in the body is the sodium-potassium pump. The sodium-potassium pump works to transfer sodium from inside the cell, where sodium levels are low, to outside the cell, where sodium levels are higher, whereas transferring potassium from the extracellular fluid, where potassium levels are low, to the cell's interior, where potassium levels are higher. The process begins as 3 sodium ions from inside the cell funnel into receptor sites on a channel protein. Fueled by ATP, the channel protein releases the sodium ions into the extracellular fluid, causing them to move from an area of lower to higher concentration. Meanwhile, 2 potassium ions from outside the cell enter the same channel protein. The potassium ions are then released inside the cell. This keeps the concentration of potassium higher and the concentration of sodium lower within the cell. Just like swimming upstream, moving against the concentration gradient requires energy, which is provided in the form of ATP. The sodium-potassium pump is responsible for regulating the volume of fluid within cells, providing the electrical potential necessary for nervous system activity, and helping in heat production.
So the big thing here is remembering that this is active transport. It requires energy in the form of ATP to move these particles across this plasma membrane. Okay, the last thing I want to talk about in this video is transport by vesicles, which again is a form of active transport, requires energy. 2 different processes here, endocytosis and exocytosis. So endocytosis brings substances into the cell. Exocytosis uses vesicles to release substances outside of the cell. So if you look in your book, these same pictures are there. You're just going to see them a little bit bigger than I kind of crammed them onto this PowerPoint. But endocytosis has 2 processes, phagocytosis and pinocytosis. Phagocytosis is what we call cellular eating. It brings particles into the cell. So that's why they call it cell eating versus pinocytosis, same exact process, except for it brings in smaller particles or liquids. So we call it cell drinking, but the same process. You can see here the particle, or in the process of pinocytosis, be a whole bunch of smaller particles come to the cellular membrane. The cellular membrane kind of envelopes or takes in those materials, becomes this little vesicle, and it's brought into the cell. All sorts of different things are going to happen to it then, depending on what's brought in, but we just want to understand the difference here, Okay, this example kind of talks about bacteria. We want white blood cells to consume bacteria. That's how we kill them and destroy the bacteria that enters our bodies. That's phagocytosis. Exocytosis is when we actually release substances outside the cell. So we have a vesicle full of substances. It comes to the cellular membrane. The cell membrane opens and releases those. One example of this is the mammary glands that are involved in milk production. So that gland secretes the milk. So just another different concept we have to remember here. So I'm going to differentiate these two discussions, even though it's one PowerPoint presentation. I want to do another recording on DNA, RNA, and mitosis, since that's a pretty lengthy discussion as well.